Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy in my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on Current Initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Kiana Aran. She's an assistant professor, part of medical diagnostics and therapeutics at the Keck Institute. And we're going to talk about CRISPR on a chip. I hope I described it properly, but I'm going to ask her about that. So, Kiana, thank you for coming.
Thank you so much for inviting me. I'm excited to be here. And it's CRISPR Chip. We call it CRISPR Chip to just make it more fun.
Well, very good. Tell me about your research, please.
Sure. Sounds great. I'm actually an associate professor now at Keck Institute, and I'm also the CSO of Cardea Bio, a company that I am a part of, as a way to translate some of my research into commercial products, which I'll talk about. So I'm an electrical engineer by training who is fascinated about biology. I always was. And I found my passion when I started doing Ph.D. and how to combine biology with electronics. So the main research theme in my lab is to use modern electronic to understand biology or to devise tools that can measure biological interactions. So biology is super, super complex. It's systems within systems that they work together to determine, our reality, our health, our diet, our physical traits, or diseases, and but also other aspects that we may not think about, such as cosmetic and health of our pets and whatnot. For years, the thought was that it would be really great if the same way that we convert streams of real-time data to digital information using the modern gadget that we have, like computers, phone, and other gadgets, can we actually convert biological data to digital information? And this was a field that I got really interested in, and it was a great way for me to connect my passion with my technical skills. Yeah, so this basically is the theme of my research, and to just give you a little history about what is it that I'm doing in the lab and what is this digital component that I'm talking about. I want to briefly talk to you about, what is the major component in the majority of the digital gadget that we use today, and how are we converting those major components to something that can really connect with biology. When you think about all of the gadgets that you work today, like your phone, your computers, there is a major element in all of these devices. It's called transistors. Transistor was one of the major technological advances of of our centuries, where it was also a Nobel winning discovery. An invention. So the transistors were invented around 1950s to address a specific, problem. Back in the days, all the communication used to happen using vacuum tubes. I don't know if you still remember, but I remember my grandfather using to have, vacuum tube based radios, right?
I've seen them. Yeah, I've seen them.
Yeah. So the communication, long distance communication, used to be done by vacuum tube. When AT&T commissioned Bell Lab to actually design something that can operate better than vacuum tubes, that you can have long distance communications in a more effective way. So this is where William Shockley and his colleagues started working on using a new material that can replace vacuum tubes. And then they discovered, or they invented transistors by replacing, by using a novel material at the time, silicon and germanium, to be able to make transistors just to address that telecommunication issues that they have. They never thought about the majority of the applications that we are using transistors for today, and those applications were never thought about at the time. It was a change of material, to silicon and germanium that enabled us to have faster switches, faster transistors, and later on we started using them in computers, and nowadays you can see them in basically any electronic gadget that you see. So silicon was great, and switching to that material enabled us to basically do that. But silicon has limitations. When we think about, okay, can we use transistors to connect with biology now? Can we expand the application of these transistors? Can we actually generate the next generation of these transistors that can enable us to connect to biology? Then silicon becomes a limiting factor. So another part of my research has been really trying to find, materials that can replace silicon. And what we really do in my lab and in a larger scale in the companies I'm involved at, really using material that can replace silicon that are able to connect with biology without impacting biology. So we use the 2D material, graphene, another Nobel Prize winning technology. That is, it's a 2D material made of carbon molecule, can talk with biology without impacting it. And it's much faster because biological events happen so fast at the speeds that, it's really difficult to capture.
What are some of the time frames, for example? Is it like picoseconds or?
Yes. Exactly. So some of these biological events are happening in picoseconds, right? And those things are really hard to capture, with optics. And this is another major challenge because the status quo today in measuring biological event, it's using optics, and as you probably are familiar with, that optical instrument are bulky, are difficult. You always have to have a label that we can optically detect, and this is because optical wavelengths are limited because they in order to be able to pass through smaller scale, biology, you need to have much smaller wavelength to be able to detect those, and optics is limited in that context. So that's why when you're doing, let's say, all of these, optical systems that we have, they all require amplification because you really have to make biology bigger and make the signal bigger in order for you to be able to observe it with optics. And this is where we are in understanding modern biology today. And there has been not much focus on really utilizing the highest speed and modern electronics to really connect with biology. So this is what we're doing. We're changing the material from silicon to something that it's faster than silicon. It can interact with biology in a much friendlier way. And we're trying to really capture this biological event in real time, and this is basically what has been enabled me and my team to be able to connect with biology and read those biological signals. And CRISPR chip is one of those examples where we were fascinated about CRISPR as a robot, as a tool that can unzip the double-stranded DNA, search through the DNA, find its target molecule, its target sequence, and then cut it, and then we can do editing.
Is anyone looking at the possible downstream effects, epigenetic change, microbiome change, et cetera? I mean, also how genes are transcribed, that affects. I mean, I know that now it's possible to target and cut and insert, et cetera, into the genome, but I would think there's a whole host of downstream effects that I don't know if anyone's looking at.
Yeah, very good question. Like, before even moving to CRISPR, explaining CRISPR to you guys. So our platform is not only limited to like CRISPR. We can really look at, interaction of, let's say, RNA binding proteins, right? Because those events are also not clearly understood. We know these things from the phenotypic result that we see from these events, right? It's really hard to understand why, for example, protein A is binding to this RNA, but protein B is not binding. Why this RNA with a single point mutation can have such a strong impact on the protein machinery.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
But then RNA-B, which has, other mutation, does not have that impact, right? So those are not really understood. And one of the things that we're trying to do, with our platform is to really look at, can we actually see what are the proteins that are binding to this RNA? Let's create some sort of a mutation and see how that mutation is impacting this binding, and can we create some sort of a stream of data that can really help us understand this phenomenon, and there is no tool that can take us to this nanoscale level and help us understand those at that scale. So this is something that it's also possible using fast speed electronic, which we are trying to take advantage of.
Yeah, I just wonder if anyone's studying, again, the potential downstream effects. I mean, I know that I guess there's a myriad of effects, but is anyone looking at that specific?
So again, one of the things that it's also lacking, and our company is trying to look at those, is that, when you look at genetic changes, you want to also look at the phenotypic result, proteomics, and then what other impacts that it has. So it really, you want to look at all the omics at the same time and have large volume of data, so you can allow computers and machine learning algorithm to analyze those. If this sort of mutation is happening, how it's going to impact the protein generation. So our platform that we have created right now, it's a multi-omics platform, which we can measure RNA binding, we can measure DNA binding, we can measure protein interaction, and the goal is really have all of those things in one system, or have one platform that can measure all of those signal at the same time and create correlations between them. And it's not only between different type of omics. When you even look at 3-4 different type of proteins together, the relationship and how those proteins and their concentration goes up and down, it's really, really important. We have no tools that can look at all of these things at the same time. I'll give you another example. IL-6, for example, is a molecule that is implemented, is a cytokine that is implemented in many diseases. It's also implemented in when we exercise or when we change diet, right? But the relationship that this cytokines, the concentration of this cytokine plays with other cytokines like TNF-alpha or other type of cytokines, and how this concentration, they go up and down together, can define, there's a lot of study that show that it can define if this elevation of IL-6 is a result of infectious disease, if it's a result of exercise. Imagine having a tool that can monitor all of these things at the same time in a longitudinal manner. Then this is a whole new game, because then you can look at the interplay between this biological molecule and understand what impacts it could have.
When you say CRISPR on a chip, I mean, is that just the omics analysis? Is that what you mean? Or what does CRISPR on a chip mean?
So CRISPR chip basically utilizes the function of CRISPR as a technology. What does that mean? One of the things that CRISPR does, it can unzip the double-stranded DNA, find this target sequence precisely in the DNA, and then cuts the double-stranded DNA, and then you can do editing, right? What I was fascinated with back when I was at Berkeley, I looked at this as a robot, and I was like, wow, what if we can monitor the function of the CRISPR as it does its search function? And can we actually use that to identify and search for a specific DNA in our sequence, basically as a genotyping tool? And the whole goal was that if CRISPR can do that in a cell, can it do that on a chip? And can we actually eliminate the need for amplification if we have a system that can precisely monitor the function of CRISPR chip? So CRISPR chip is basically a genotyping tool where you can detect your target DNA without the need for amplification. So you just take your genetic material, you put it on the chip, and our chips are decorated with CRISPR, and then CRISPR can do whatever it does in the cells. It looks for a target, bind to it, and then once that binding happens, our chip can detect it.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And we have been able to detect multiple different diseases using the technology. We have been able to detect sickle cell disease, ALS disease, Duchenne muscular dystrophy without even needing to amplify the genome and directly detect the sequence of interest. So this is what CRISPR chip is as a gene search engine, we can call it, where we actually eliminated the need for amplification for some diseases, mostly genetic diseases so far, and we're expanding it for other applications as we speak.
Why don't you need to, when you say amplified, you need to have many thousands or millions of instances of CRISPR in order for it to work? Is that what you mean by amplification?
Yes, so not CRISPR, but your target. So the main way that DNA and sequences are detected today, even when you're using next-gen sequencing or other type of sequencing technology, you usually know what target you're looking for, and then you design primer where you really amplify your target to thousands and millions of target molecules before you can detect it with PCR or sequencing methodologies, and this is a major bottleneck because PCR is very complex. It requires specific lab facilities, and in order for us to be able to just amplify the target before we can even detect it, and it's major limitation that we try to.
But how many CRISPR molecules are needed to accomplish a given break and repair? How do you know the ratio of how much is needed to the number of cells to be affected?
Those we test, those are all done with in vitro testing. So right now with 1,000 CRISPR molecule in our chip, we can detect, easily genetic mutations that from DNA material that are, obtained with a single buccal swab. So if you have a single swab and you take the genetic material out of that single swab and you put it on the DNA on our CRISPR chip with 1,000 CRISPR on it, we can easily detect if search those mutation and detect those. Of course, for other type of diseases like infectious diseases where the target material, where the amount of DNA is not that much in the system, then we need to, we are trying to improve the sensitivity of our platform to be able to also detect those without amplification.
So the genetic material, when working with CRISPR, what does it typically take? Millions of replicants, or, how much savings are you getting in terms of replication amount?
So when you, in a single buccal swab, you have about 20 to 30,000 molecule of DNA molecule, and that's enough for us to be able to detect, provide a yes or no answer if you have that specific target gene in the sample or not. Normally when you do in normal processes, you have to create millions and millions of that target in order for you to be able to detect it with optical techniques.
Are there parts of your research that are now in clinical trials, or how far along are you?
Yeah, so perfect. A great question. So the CRISPR chip technology, we call them a CRISPR chipset. They're part of the company I'm involved at called Cardea Bio. We're actually working with partners for various different applications of our chip. So the first application of our chip, it's actually not detecting genes for genetic diseases, but using it for CRISPR, quality control application. One of the major challenges with CRISPR technology is that, we haven't been able to effectively or master the use of CRISPR. We still have to assess CRISPR efficiency in using some sort of an in vitro method that are very lengthy, they're not very accurate. So there's a lot of effort on ensuring the efficiency and the efficacy of CRISPR itself. So we have many partners we work with to identify which CRISPR design it's best suited for their application. If they're doing editing, how much their editing has been successful. So before you move to cell trial or to in vivo studies, you can test for various different type of CRISPR molecule and identify the ones that works best for your specific application. So this was the first application of our technology that we offer to the market, and it's a commercial service that is available for that based on this technology, which we don't have to do any amplification of your sample. You just you edit your cells, and then you look and you monitor the editing efficiency before and after editing, and then you compare it across different type of CRISPR and RNA complexes, and you see which one is more efficient. We're also doing a lot of genotyping for plants as well, where you have people in the field where they want to see if their plant are modified or not modified, and they can also use CRISPR chip in the field to do those type of testing, and other applications which we work with our partners to use the technology with. And of course, we have to improve the technology for infectious disease to be able to reach a point where we don't need amplification for any of those applications.
Why is there amplification not needed? Is it like a microfluidics flow system on the chip that allows you to use a lot less material, or what are the factors that allow you not to have to amplify the material?
So our chips, because they, as I mentioned, they're made from graphene. Graphene is very, very sensitive to charged molecule. That means if a charged molecule comes close to the graphene, it can change the conductivity of the material significantly, and that can be detected easily with our system. So now imagine you have a CRISPR molecule sitting on your sensor, and it interacts with your target DNA. If it binds to your target DNA, it brings the whole DNA down very close to your surface of the graphene. And because DNA is charged, it's a negatively charged molecule, it impacts the conductivity of our material, and therefore what the transistor is reading in terms of current and voltage. So basically we are using the property of biological molecules that are charged, because proteins are charged, DNA is charged, and this charged molecule can impact the graphene conductivity, and that we can sense very sensitively with our chip. So now having a single DNA molecule, if you bring that down closer down to the graphene, that can impact. So now we have 1,000 CRISPR sitting on the graphene, so that they potentially can bring 1,000 molecules down to the surface, and those change in charge can be detected.
Okay, I see. So in most lab systems, and then also in biological systems, there's so much going on. There's so much material moving around that the efficiency of CRISPR is just way lower, right?
Yeah, and people are not using really CRISPR for DNA searching. The majority of people are using CRISPR for editing, right, in cells. So we're just combining basically the power of CRISPR in searching, because CRISPR basically hops or scans the whole DNA to find its target. Whereas other techniques where they detect DNA, they really have to have a probe specifically designed to that specific piece of DNA. That means that you have to create a lot of those pieces of target DNA to enable those interactions. Because if you don't create those small pieces of target DNA, then those interactions are very difficult to really happen. You have to have abundance of those target molecules for those interactions to be able to happen effectively. For us, we don't do that. We let CRISPR does what it does, and we're really using the power of biology as technology rather than trying to reinvent the wheels.
Okay. What do you think is going to be possible with your research and similar research over the next few years versus, what's going to take maybe a decade or two?
So I think when we designed the technology, we really were thinking about, the genotyping capability of our technology, but then we didn't realize that, throughout the process of, putting the CRISPR molecule on the chip and introducing the RNA, we were actually creating a tool for quality control application. What does that mean? A lot of time when people are using CRISPR technology for gene editing, they have to make sure that they have the right RNA molecule. They have to make sure that some modification that they do with the Cas enzyme or the RNA molecule is not impacting the complex. And the beauty of our technology is that you can put the CRISPR on the chip, then you can pass your RNA molecule, and then you can see how stable the interaction is, if they're binding, if there's anything impacted that. And then you can put your target DNA or target RNA based on what you're trying to do, and really look at those interactions in real time and modify them. So what we didn't really notice that we were creating a perfect tool for CRISPR quality control application. So that's the first thing I see in future in terms of CRISPR. But is CRISPR the only thing that can be done with our sensor? Absolutely not. There is so many other biological molecules that have fundamental functions that we are able to capture those functionality on our chip and create really and basically utilize biology as a technology on our platform. Example of those, olfactory receptors, they are very sensitive to small molecule, and you can smell things basically based on those. What if we put those on the chip? You can create an electronic nose. And in fact, we have, our company have signed a new contract with one partner where we are, designing those electronic nose using this biological molecule. So another example of using biology as technology. And again, in general, because the capability of our transistor, because they're fast, they can monitor a lot of biological interactions, and there is no label, there is no optical labels. You can use them for monitoring protein-protein interaction, protein RNA interaction, protein DNA interaction, and a lot of this thing can be done by simply reconfiguring what you're putting on the chip. Whereas in status quo, like ways of things that are being done right now, the status quo for that is that basically you optically label everything, and those labels cannot be the same. You have to design and spend a lot of time on designing those labels. So the reconfigurability of our technology and the way that we can scale it up for different applications, I think it opens door for many new findings and exploration for biologists and in looking at these biological molecules and biological events.
Well, very good. Kara, what's the best way for people to find out more about your work? Where can they go?
If you guys want to look at our company, Cardea Bio, where we are, commercializing basically a lot of this transistors-based technology for interacting with biology. And also my lab at aranlab.org, where you also can learn about what other things we're doing, using electronic for biological application and improving our biological understanding.
Well, very good. Kara, thank you for coming on the podcast. I appreciate it.
Thank you so much.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.